Now we're going to move from design to execution.
It's the #1 clicker. Okay, good. All right, hi everyone. I'm Kristin Ellis. I'm the Director of Scientific Development at OpenTrons, and we make robots for biologists. Let me make sure that... I was holding it the wrong way. So today I want to talk to you about the fact that biotech is still handmade. The vast majority of research labs don't have any automation, and the scientists in those labs still run all of their experiments by hand. Human error is costly, as was demonstrated by Len in his talk that he introduced this morning, and introduces a lot of variability. And this is a major contributing factor to irreproducibility in life science research. And the reason that most biotech is still handmade is that laboratory automation still exists in the mainframe era. So like computers in the 60s and 70s, today's lab robots are very expensive, very difficult to use, and the technology is closed to most people. So while sophisticated biological tools like DNA synthesis and sequence data have become more accessible in recent years, sophisticated laboratory tools like lab automation haven't. So at OpenTrons, we like to ask ourselves the question what it would take to get automation into every laboratory. So what if we could put robots in all of the academic and industrial labs that to this day still automate everything by hand? We think the answer looks a lot more like a PC than a mainframe. It has to be affordable, easy to use, flexible, and open. And what you can boil all of those things down to in a nice little package is accessibility. Because we have to remember when we look back at the personal computing revolution, the acceleration in innovation happened as computers became more accessible. More people, and more importantly, even more types of people, could access and use computers. They became less expensive, and the user experience became better, so they required far less specialist knowledge to operate. We want to apply those ideas to the paradigm of laboratory automation, providing a platform for executing consistent and reproducible workflows. So we currently have more than 450 OpenTrons robots in dozens of countries around the world, and we've been doubling our user base every 6 months for the last 3 years. I won't be able to tell you the stories of every single person on the OpenTrons platform, but I do want to relate to you some highlights from our user community and tell you the stories of a few biologists that have integrated our robots into their workflows to increase the reproducibility of their work, starting with the Mayo Clinic. So Dr. Ecker's group at the Mayo Clinic is one of our earliest collaborators. They actually bought an OT1 through our Kickstarter campaign, and they published the first paper on OpenTrons in the journal Human Gene Therapy, where they described a novel way to construct talons called FuseX. Not only did they publish those results, they also published the source code for the experiment that they run on OpenTrons, so now anybody can download and run that same experiment and reproduce their results. So together we've created a push-button run experience for a gene editing workflow. So using OpenTrons hardware and software and the Mayo Clinic's FuseX kit, users can type in their 15 base pair target sequence, set up the deck and hit run, and they can assemble their own custom gene editing protein. This is scientist Kaya Wasik at GenCove. She's the chief scientific officer, and she uses OpenTrons to automate their whole genome sequencing pipeline. So when you're dealing with customer samples, there's absolutely no room for error, and there's a high risk involved with irreproducible results. So everything has to be done to a high degree of accuracy, and operator mix-ups are not an option. So before OpenTrons, a startup like GenCove would not have had access to the tools to automate their whole pipeline. So we certainly brought down the cost for them. And flexibility is also a key. So a scientist like Kaya can do a few programming changes, manipulate the code a little bit, and assemble their samples with higher efficiency, which allows 2 technicians in their lab to process up to 500 samples a day, generating high quality data every time, and making it without errors. Now, Willats is an interesting use case. They're a startup that exists both on the West Coast and on the East Coast. The bulk of their R&D happens here on the West Coast in the Bay Area at the Oakland Genome Center. And the bulk of their patients' sample testing happens on the East Coast at the New York Blood Center. So this means that their core workflows are being developed in one place and they're being executed in another. So without automation, that is a disastrous recipe for irreproducibility. So they've started to automate a lot of their processes on OpenTrons, including nucleic acid purification. And most recently, they published a process for massively parallel qubit DNA quantification using OpenTrons. So they published the source code on OpenTrons and the SOP and run notes on protocols.io. So their technicians on the East Coast can run the exact same experiment as their technicians on the West Coast. So you have two labs 3,000 miles apart with multiple operators, and it's a perfectly reproducible and collaborative effort with no error. So open robotics has been the key to getting that result. Now, a falling price tag is not the only thing making our robots more accessible to scientists. Our platform has allowed people to reimagine how humans might interact with robots, further lowering the barrier to entry. So this is one of our favorite examples.
Alexa, tell RoboBench to make a 1 in 10 dilution series with 5 tubes.
Okay, making a 1 in 10 dilution series with 5 tubes.
Okay, I don't have time to run the whole video, but that's awesome. That was prototyped in one afternoon by Anton Jackson Smith, who's a graduate student in the Indy Lab at Stanford. So that's the true power of an open source platform. Anybody with some programming skills and a lot of imagination can prototype anything they dream up, including a way to tell your personal lab robot to do your bench work with a voice command. That not only increases accessibility for people with great imaginations, but it might also increase accessibility for people who are differently abled in the lab. I'm going to wrap up really quick because I'm getting the stop and start Q&A. So that's what we serve to do. We want to lower the barrier to entry for lab automation and provide an entry point into the world of sophisticated biotechnology tools that was prior to this completely inaccessible to the vast majority of the life science market. So with that, I'll take some questions.
Hi. Question about the Alexa integration. So someone over this weekend at Science Hack Day in San Francisco at GitHub started integrating Alexa with protocols.io, which is very exciting, and you just had a beautiful demo of that for OpenTrons. But I'm thinking from the morning's presentations, will there be issues with vocabulary and the science-specific recognition of terms that we're going to run into?
I think, yeah.
Whatever keywords you want Alexa to learn for.
Well, Sean answered the question, but I mean, I was going to say, I think that's absolutely a possibility, but that's one of the beauties of leaving the platform open source and integratable so that we can actually troubleshoot and solve for those problems as they come up.
So traditionally, academic institutions haven't been great partners in terms of pursuing reproducibility and have not contributed a great deal to date to the discussion. OpenTrons has had an interesting experience with an academic institution that actually chose to purchase your instruments and install it across the university in different labs. And I was just wondering if you might share that story so that we can benefit and understand how academic institutions can actually be driving reproducibility.
Yeah, so that was actually done by NTNU in Norway, and so that was a pretty big contract. It took us a while to get into that university, but ultimately we ended up sending 12 robots, and we went over and helped them get set up and trained. I think those larger contracts across large universities might be a little more difficult to pull off, but people are certainly warming to the concept. Really, the bigger advantage is providing the automation to the single labs, so that then they can share those workflows, between individual labs instead of across an entire institution, and even globally. So I think that's, I don't know if that answered your question.
So I'm assuming that the institution allowed individual labs at the institutions to use it, right?
Yes, absolutely.
So I'm missing the connection between it's better for academic labs rather than institutions themselves.
I wouldn't necessarily say that that's the case. No, institutions that want to have collaborations on interlab measurements or interlab reproducibility studies could certainly benefit from having multiple of these in their institution. Okay, I don't think I can take any more questions, but we have a booth in the back, so feel free to come talk to me after. Thank you.
Good afternoon, my name's Yvonne Linney. I'm the CEO of Transcriptic, and we are the robotic cloud lab. A lot of the discussions I've heard this morning already revolve around both reproducibility, around turning biology into a data technology, which really it is, and enabling the biologists to really focus on the data and improving their experimentation, rather than focusing every day on moving little liquids in tubes around the lab. So we've taken the approach of very much trying to help the biologist very much focus on the data moving forward. So I want to tell you today about discovery biology on demand, and we've taken a lot of the tools that we've heard about today in terms of being able to code up your experiments and using IoT technologies all together, and of course robotics, all together to produce the remote biology lab. So we've heard about this problem several times already today. Both life science research is extremely capital intensive, especially if you want to go into if you want to build an automated laboratory yourself. And technically, very often it's very unreliable as well. We've spoken a lot about reproducibility, meaning results across time can't be assembled into true data sets, which is a key problem in the industry, and as we've heard, extremely expensive as well. And then finally, sharing methods with a partner can take weeks and thousands of dollars, especially to get it right. So I'd like to introduce you to the lab of the future and the lab in the cloud. So we're really trying to change the paradigm of the way biology is done by enabling access to our robotic labs directly from your laptop. You can run bioassays on demand from anywhere in the world, on the beach, in your living room, at dinner, wherever you want to be, and monitor the progress in real time and get data as soon as it's complete, speeding up the whole process and making it very reproducible. So collaboration has always been a problem, but just like collaborating a shared document remotely, as we all do nowadays with Google Docs, etc., you can now do that with biology using the Transcriptic Cloud Lab. You can really collaborate from anywhere in the world, from the comfort of your laptop or the convenience of your laptop. And as long as we've got your protocols, you can run the assays remotely whenever you need to. And this whole concept seems to be taking off. Recently, Lilly announced that they built a brand new facility in San Diego, and within that facility, they are enabling remote access to drug design and synthetic chemistry. And I think it really is sort of one of the ways of the future. So we're excited to see, without obviously using Transcriptic, that they are also thinking about the same processes. So how does it work? Everyone asks us that question when we say we have a robotic cloud lab. So the first thing is that obviously we need samples. You can't run any biology without any samples. And so we've got a very simple process for you to send your samples to us once you've signed up for an account on the system. And then we use we obviously run all your protocols on our robotic systems. And so the thing that is really important before you can get going on that as well is we need your protocols, and we convert those protocols into code using our language, or it's actually an open source language of auto protocol. And our scientists spend a lot of time initially in the implementation process working with the scientists to make sure that those protocols are very robust. They're tested routinely, both with water samples, and then looking at or using controls to ensure that you've got a very robust process before you start with your real samples. And then once everything's set up and you're trained to use the system, which is extremely simple, I'll give you a couple of examples in a second, then you can control those your protocols whenever you want to, launch a run, and get the data as soon as those experiments are complete. We can run a variety of different assays on the system today. Here's just some examples from a bioassay standpoint, using either biomarker assays, cell-based assays, or biochemical assays. Our menu is continually growing. But really, we focus at the moment on any sort of plate reader, qPCR machine, fluorescence, luminescence, etc. So let me just give you an example then of how to run an experiment through the internet. This is using MSD, which is the mesoscale detection system, as an example. So first of all, you as a researcher have an individual, your own project board, and this is what your project board would look like. And so you select from your project board, you have a series of protocols within that board. So we said we were talking about MSD today, and you can see there's an MSD assay on that board. And so if I click on that particular card, I will then get the details of that set up there on the screen. And so then there's some pull-down menus in terms of choosing which of the kits for MSD you would want to use, give that experiment a unique name, select your samples, which you do through your samples that you've already got in inventory. You can select individual ones of these samples in this 96-well plate, or you could select them all very, very easily using typical tools that we use every day in other things. And then finally, you would basically validate that run to make sure you haven't made any mistakes before you start. So the system will automatically validate the run, and then it'll be ready to launch. So once you're ready to launch, you basically press launch in the top left-hand corner there, and then, I don't know what's going on here. Oh, okay. I should actually realize this was live. Okay, so you're just looking at details here that you can scroll down on the left-hand side. This is while it's running. You can look at the various different parameters that you've set and the way your experiments are running. And then at the end of the day, you will see your results come up as your plate map here, looking at different analytes and looking at the results in the different wells, depending on the various analytes. So at the end, after you've run your experiments, you can basically download those data from CSV files into your favorite analytical software, and so you're ready to go. So I've been told that I need to stop, so I will actually miss the last part, which I was going to give you an example, but hopefully the slides will be shared anyway. But I think I've given you an idea of what the Robotic Cloud Lab looks like. This is just showing a lot of data that we collect through IoT tools that we use, so that, again, we've all been talking about variability and whatever. So if you see some variability in your data that you don't expect, you can do a deep dive, again, remotely, to look at the various parameters that we're measuring to see whether you can see the cause of that variability, or whether it is actually a piece of data, a data point. So with that, I will stop and open to questions. Thank you.
What kind of reports? Every time, sorry. What kind of reports do you get from clients about? Do they do a lot of tests of the system and the variability? Do they giving you feedback about how it compares to their experience?
They haven't necessarily given us sort of feedback on their general experience, other than very, very positive. I mean, people who use us are really, really impressed with the data that they get back. So I'm not sure exactly how they're comparing with what they've done it before, but certainly in terms of enabling their scientists to really be able to focus on experimental design, and then, they can go on to the next experimental design whilst everything's running.
Where do you see sources of variation with these robots and this kind of automation? Do you? Yeah, I mean, you must. It can't be perfect.
Yeah, I mean, obviously, sort of liquid handling is always a challenge. And so I think, we use off-the-shelf liquid handlers. And certainly, sort of, there's still challenges in that area that we monitor very closely.
If we zoom out, there's sort of going forward in the future, there's two pushes that we're seeing even at today's event. One is bringing automation into the lab, into the hands of the scientists doing the experiment, and one is bringing the sample out of the lab to something like Transcriptic or Emerald Cloud. 10 years or 20 years from now, how much of the science that we do you think is automatable and can be outsourced, and we just send the sample and the entire thing happens, versus how much needs to be automated inside the lab directly?
I think it very much depends on what the actual sort of life cycle of that sample is. So I would say that in very routine assays that are run sort of every day, then the outsourcing is very, very simple and very sort of in demand. I would say that where you've got proprietary products, et cetera, then that's when you want to keep it in-house. So I see that split as the two. I'm being told to end, so pass it on to the next person.
One or two.
Okay, great. Thanks so much, everyone. My name is David Sun Kong. I'm the director of the new Community Biotechnology Initiative
At the MIT Media Lab. I just wanted to thank Nancy and Will and Doug and all of the wonderful organizers for putting on an amazing event. So I'm going to talk to you today about an area of research I've been working in for a while called open source fluidics, and as applied to synthetic biology and biotechnology in general. We've been learning all morning today about different types of technologies used for engineering living systems. Others have presented kind of similar slides reflecting on technology and maybe how far we still have to go. This is the first pipette patent, and over 100 years we're still manipulating fluids in a pretty similar way, which is pretty remarkable when you compare it to information technology. I work in a field called microfluidics, which has a lot of promise, I think, for trying to automate and create more higher throughput, more reproducible systems for bio. So again, miniaturizing large-scale biological processes into chips. These are examples of some systems where we use, for example, silicone rubbers like polydimethylsiloxane, which can be used to create automatable pumps and valves for kind of metering and controlling fluids at very tiny scales. So again, you can make automatable mixers. And again, the typical volumes that we work with in fluidics are nanoliter, 10 to the minus 9 liters, sometimes even smaller to picoliters. And so, with some of these kind of very precise technologies, there's been a lot of work done in almost 10,000 papers in microfluidics. I'll give you a couple of quick examples from my own work. This is from my doctoral work, where we took the outputs of DNA synthesizers, so short chemically synthesized DNA oligos, and then assembled them into longer gene length constructs in microfluidic systems. Over the past couple of years, before joining the Media Lab, I worked at Lincoln Laboratory at MIT, and we worked on developing these kind of microfluidic systems for miniaturizing genetic circuit assembly, so building large-scale DNA constructs, thousands of bases in length. And this system in particular is an open hardware system where kind of the tools are all built off of Arduinos and other very accessible commercial off-the-part shells for just hundreds of dollars. And then, again, we miniaturized a lot of these common assembly biotechnologies into microfluidics and showed that the stuff we built on chip performed as well as the stuff that was made in kind of conventional tubes. And then again, one of the great areas about fluidics is the possibility of doing high throughput experimentation, scaling up to tens to hundreds of different types of experiments in parallel. We'd love to move to a scale of being able to do, say, like 100,000 things at once. Very challenging, but that's the dream. Other examples of some high throughput fluidic systems are droplet fluidics. Again, many of you are probably familiar with these. So this is kind of an example of some simple droplet microfluidics where we're encapsulating fluorescent bacteria, which we can then assay. You can also make very kind of monodispersed high throughput droplets. So you can make these guys at a frequency of around 1,000 droplets per second. So you can really achieve very large scales in terms of the types of fluids that can be manipulated with droplet technologies. And so quickly, I'll move through a few examples of some applications. One collaboration that we've worked on is one with Professor Ron Weiss's lab at MIT, where we worked on building genetic circuits that could be used as classifier circuits for identifying and destroying cancer cells. So the basic idea is that if you can monitor the concentration of just a handful of microRNAs in a cancer cell, then you can selectively identify that as cancerous as compared to a healthy cell. And then Ron's group devised these kind of really cool genetic circuits that can kind of ask a digital Boolean logic question of the cell, kind of querying the concentration of a few key microRNAs. And then if you pop these guys into a set of cancer cells and healthy cells, then it'll differentially kind of kill the cancer cells. And so we've been working with Ron to build these kind of large-scale fluidic systems to prototype hundreds of these different types of sensors in parallel. Another example is in protein engineering. So again, one of the big areas for protein engineering that's pretty exciting is bioremediation, so being able to use proteins to break down toxins. And so again, with protein engineering, one of the big problems is, how do you test all of the different protein variants? And so if you've got really good in silico tools, you can simulate them, but also, of course, it'd be great to be able to just build lots of versions of these in parallel. So for one particular project, we worked on trying to make many variants of a type of enzyme called organophosphate hydrolase, OPH. And basically, we use this high-throughput chip to introduce cell-free extracts, which I think we're going to hear a little bit about later, which I think is a really powerful enabling technology coming up. And the idea here is to print on the chip these different kind of DNA variants of this protein, and then flood the chip with the IVT, and then using fluorescence to kind of detect and assay each of these different designs. And so with a type of system like this, you can kind of extract data from, say, 100 different proteins that you're manufacturing all on the surface of a chip. And then as a final quick example, again, the human microbiome is a really exciting area of research. We've done a lot of work now recently kind of studying different ways to construct synthetic microbial communities. And again, I won't belabor the point, there's a lot of relationships between the microbiota, particularly the gut, and almost every system of the body. And what we did was to evaluate some of the most kind of advanced 3D printing techniques and looking at ways to use these 3D printing tools to make kind of physiologically relevant systems. So leveraging against advanced gradient and kind of bitmap printing tools to make these structures. And this is a collaborator, Nuri Oxman from the Media Lab, who's made some beautiful 3D printed fluidics. And then at Lincoln, we worked on making these kind of multi-material structures that can do, kind of peristalsis and simulate some of the crypt structures you might find in a real gut. Again, to prototype microbial communities. And so ultimately, the big question is, how does open source fit into this, and how do we share all of this stuff? And so this is a big paragraph, but I'll read you just the sentence. So open source hardware is hardware whose design is made publicly available so that anyone can study, modify, distribute, make, and sell the design or hardware. So basically, how do we share designs? How do we build chips out of really common, easily accessible tools? At MIT, I've taught a few classes on open source fluidics where we, kind of used commodity 3D printing tools, did DNA assembly inside them. And again, with 3D printing, you can kind of rapidly prototype through the design cycle really quickly, hours instead of days, and make some really cool chips that you couldn't make using standard 2D methods. So this is an example of a three-dimensional micromixer where we are doing a genetic circuit assembly inside this fluidic device. And Again, partially with from the classes, we were able to publish some really neat papers. I should point out here, this is Julie Legault. Prior to just a couple of years ago in this class, Julie was a designer, had no experience with biology, and she's now the CEO of Amino Lab, which is a really cool kind of tabletop culture system. And this is Will Patrick in the back, again, also a designer, no previous bio experience, and he now is also the CEO of his own bioreactor company here in the Bay Area. So I think we're kind of entering this really amazing phase where there's a lot more kind of creative folks coming into bio and, without fancy degrees or a lot of training, really able to make an impact. And so just to conclude, we're sharing all of this stuff. I'm going to skip through here because I just got the must stop sign. We're sharing all of this stuff via Metafluidics, which is this kind of in a way, sort of like a GitHub or a Thingiverse for fluidic parts. Again, for those of you that work in microfluidics, you know that the papers that you publish in fluidics are kind of like the tombstone of your work. Like, after you publish the paper, nobody ever reuses your chip again. And so we're trying to solve that problem by creating this open repository where you can share all of these fluidic designs. So, with the whole goal, ultimately, of enabling lots and lots of diverse communities all around the world, including folks like this, which, we had a meeting, again, Bethan mentioned this, where we had a couple hundred folks from all around the world who are setting up independent community laboratories, and they all came to the Media Lab. And again, I think it's a really exciting movement of new communities that are participating in bio and really helping to impact the future. So thank you very much. And sorry for talking so fast. That was like rapid fire. But any questions?
I'm interested in your perspective. What kinds of things are perfect for microfluidics? What kinds of things are hard? What kind of things are terrible for robots but great for fluidics? Like, how do you see these things working together?
Yeah, super great question. So, again, I didn't mention this clearly in the talk, but one of the major benefits, obviously, of fluidics is just minimize reagent costs, right? So we're talking about being able to do stuff at nanoliter or sub-nanoliter scale. So, again, I think there was a talk earlier today that really beautifully kind of talked about reagent waste and so on, and that's one of the big sweet spots, I think, for fluidics. It's a good question, what's good for fluidics versus robots? I think microfluidics in general, there's lots of reproducibility issues with that technology in general. So, I think with tools like an infrastructure like Metafluidics, I think we really need to reproduce more fluidic chips and take that cool science paper where somebody published a cool thing and actually show that this chip can work in many different settings. So I think part of it, there's a lot of technology evaluation that needs to happen to really make that comparison more strongly. Yeah, so I think at the end of the day, both of those tools can totally work together in concert in powerful ways. So, but I do think there's a lot of evaluation that still needs to be done. Okay, thank you all very much for your time.
This one? Number 2. Hi, I'm Michael Mermelstein. I'm here with Cam Abnett over there we're from Umech Technologies. We're an instrument company that you've probably never heard of. Most of our products get rebranded in a kind of OEM situation. I'm going to tell you about some work that we're doing now. Closer, does that matter? Originally funded with some support from the NSF to connect biologists with automation. And in particular, I want to tell you about a language called BioIO. Try this button. Nope. Try this button. This button. No. Hey, help me out. Oh, here we go. I got it. Sorry. All right. Okay. So this is who we're trying to help out. This is a bench researcher, maybe a familiar scene. We had an opportunity to follow Monica here and a bunch of other people for a couple of days and observe this customer, this archetype user, and what her problems are. And we observed some commonalities. So here she is working at a crowded lab bench that's probably familiar to a lot of people here. She spends a lot of her day doing pipetting manually. Even though she actually has access to a beautiful Hamilton robot right around the corner from her bench. But this is more efficient for her. So how can we help her? That's our job. How can we connect her with automation? So here's a sort of product vision for personal robot for her, right? It's a small thing about the size of a stapler or one of those big tape dispensers. It's $900. So it's personal, right? And it fits in the drawer when she's not using it that she keeps the rest of her pipetters in. So you can see, let's see if I can, yeah. So it drives around on the edge of this Tupperware tray and draws up liquid from wells of 96 well plates. So I asked, so what are we going to do with this thing, right? So I asked Monica, like a second after taking that previous snapshot, what are you doing? And she showed me this. This is exactly, in fact, what she was doing at the time of that previous snapshot. She's running a kit, right? She's personalized the instructions from this kit, right? And so that's our job. We're going to try to do this type of work in that type of environment. So we need something that really fits into her workflow, into the sort of reality of what she's got to do to succeed, right, in her job. So we looked at a bunch of ways of connecting this idea to our personal robot vision, like programming languages, visual languages. There's been a lot of talk about like drag and drop interfaces. We looked at a lot of paradigms. So how are we going to capture this into a language? And where we ended up in our brainstorming was that, look, it's already in a language, English, right? This is a language that she already knows. She doesn't have to learn anything. So that was our idea. So how far can we get just interpreting English sentences like this, right? And that's really what BioIO is. It's not exactly 100% right. So BioIO is not the entire space of English sentences, right? We've created a reduced grammar. We've restricted which sentences make sense, right? So by looking at maybe a few thousand protocols from kits and from journals and from protocols.io, we sort of found some common idioms that appear in a lot of protocols, and we've refined those and written kind of a grammar book for like what kinds of sentences are valid BioIO speak, right? And by doing that, we have some benefits over doing like the normal natural language processing problem, right? The advantage, like the first advantage from sort of a computer science point of view is that we have a deterministic parse of the sentence with an unambiguous meaning, right? It's the interpretation is defined by our grammar book. And so that allows us to create much more easily an interpreter that deterministically schedules low-level operations for our robot or other automation. So you can see here, like, so this is just an editor in a web browser. That's our sort of prototype that we use for interviewing a number of researchers that we have access to. Like you can see a few things about it, right? It's English. It's English. So you can publish your protocol exactly as you write it for the robot, for one thing. Another thing is, it's a little subtle. So like the first step in her, like if you go back to here, like the first step in her protocol was the liquid handling step. And the second step is this weird step, apply vacuum, which is not part of the grammar that we've defined so far. It's an unrecognized sentence, but like unlike most computer languages, we're not going to have like a syntax error and complain to the user, right? It's just, there are no errors, right? It's just not recognized. And so that sort of reflects the reality of this kind of work. There's going to be a mix of manual and automated work, and some things are just not going to be handled by our robot or by some other piece of instrumentation. And that's going to sort of the idea of like taking your pipetter and putting it down or putting it in the drawer and doing what you need to do to sort of finish your work. That's part of it. And then also, you know, in sort of 2017, when you type text into a web, you know, editor on a web page, you know, that enables a lot of things like sharing and versioning and all the stuff you would expect when you do that. Yeah. So I want to, this is the concluding slide, actually. The big picture of the project is I'm trying to connect the bench biologist, the researcher, to automation in a way that works in her existing world, right? So I'm not trying to get us to something that's 20 years from now, or 10, or whatever, or 5, or 1. This is today, and what can we do to help her out? And so connecting her to automation, helping her record and communicate with equipment as a side effect also can help her share her work with others and communicate with her community. And then sort of on that topic of community connections, we're sort of early in this project. And so we invite collaborations if you're in any of these sort of 4 boxes, if you're a researcher who wants to try to work with us, if you're a CS person, a computer scientist who knows how to write interpreters or who knows how to write solvers and schedulers and so on, we want to collaborate with you. If you're a developer of automation software, then we can target your software as a sort of a back end for this interpreter. And finally, if you're a vendor of automation hardware and you want to work with us, we're open to that collaboration as well. So I can now invite questions.
This looks really awesome, and I think it's a piece that's been missing for a while in this ecosystem. I'm curious what your sort of business model or plan for getting it out there. Is it open source? Is it a software you buy?
Yeah, that's a great question. Our business model is to sell robots. I mentioned before, it's a $900 robot. I want to make that really popular and sell a lot of them. The software, for people to adopt it, it should also target other people's robots. So we're not going to make PCR machines. We're not going to make, we're making a liquid handling robot. We want to make that work, but we want the user to be in an environment where everything that they need to get done kind of maybe fits into this world. So we're trying to make that side of it more open and connect to other machines. And in terms of like the end game, probably where this will end up is that a partner company with a well-recognized brand will take over the project at some point. And so we have to be careful about what we do with it at this point, because we don't know what the interests are of that other company. But for now, as scientists, we want to collaborate with as many people as possible. Thanks.
Scientific research changes the world, from breakthrough technologies to life-saving medicines. However, scientific research can be incredibly slow, and it can be incredibly costly. My name is Ignacio Willats. I'm a co-founder of HackScience, a startup accelerating research with affordable and configurable lab automation. So this is Dr. Michael Delves. He's an incredible malaria researcher at Imperial College. However, Michael has to spend up to 3 hours every single day replenishing the media for his parasite cultures like this. And it even has to come in on the weekends. Unfortunately, Michael's not alone. The average researcher spends at least 50% of their time in the lab on manual processes. Now, this costs the world over £25 billion every single year. Now, further to that, as we've already discussed, this manual work really contributes to the reproducibility crisis because of the way we distribute and write down those methods and how people interpret them. So wouldn't it be amazing if all this work could be automated? Well, in Michael's case, you can. You can go out and buy one of these kits for £250,000. But let's be honest, Michael and most people out there can't afford this. And look at the size of this thing. It's not going to fit in Michael's lab. Now, unfortunately, this is a trend across lab automation instruments. They're big, they're costly, and if you look at this one, like this guy has to be there and babysitting it. So is it really automation? So that's why we built CellFeed. CellFeed's the first compact and personal solution to automating cell culture. This guy can automate the media replenishment of your cell cultures, passaging, and you can even monitor your cultures remotely, so you don't have to come in the weekends anymore. So what we're hoping to do with this is bring down the cost of doing cell culture, but also reduce the time people have to spend on this activity, because as many people have told us, this activity really doesn't provide any intellectual value to them. So we want to relieve scientists of this pain. And because it's a closed system, we can reduce contamination. We're excited to announce that our beta program is now open, We're being trialed at labs at Imperial College and UCL, and we're supported by some great organizations. But if we look further ahead, I know what you're thinking. There's more to research than just cell culture, right? And that's exactly what we thought. And we looked at the problem, and there's a few parameters that you have to consider when looking at this. Research changes day to day. You have new ideas. It changes, you change the way you work. And people don't work in the same ways, and you're constrained by a number of things. As we know, cost is one of them. Space is also another one of them. And the usability of technology has to increase. So in the future, we want people to look at the old ways of doing things, but instead of grabbing a pipette or drawing out how they're going to produce these protocols or new experiments, we want them to grab some interoperable hardware modules. So this is the idea that we've got at the moment, which is the HackScience platform. It's a suite of interoperable lab automation modules that people can use almost like Lego to prototype new workflows and new experiments. And we're making it incredibly easy to use with this interface. We know we've seen a lot of this today, a lot of drag and drop user interface and workflows, and it's the approach we're taking. So if you want to chain these different modules together, you can easily drag over these nodes and connect them. Almost think of them as data flows or ways you can configure these modules to start thinking of new ideas. And with this, you can create these really simple user interfaces so you can control your experiments. Now, the interesting thing about this is you change the paradigm from writing out your method and someone else interpreting it a completely different way to using something like this and deploying it to your hardware and just sharing a link in your research paper so someone else can deploy it onto their hardware. And you've reduced that whole challenge of trying to interpret a written method. So we see this as a potential way to really tackle the reproducibility problem in science today. So you might be asking what kind of modules might this include. So we're looking at things like syringe pumps, we're looking at things like microscopes, as well as a variety of different sensors. And we're really interested in applications like microfluidics and that kind of thing. So I'm not doing this alone. This is the incredible team that I've got supporting me. Ali and I met each other at Imperial College, and most of us studied there. And together we're really trying to push the envelope in this area, and we're really inspired by some of the things that people have demonstrated here today, and we're excited to be part of this movement. So some of the challenges we really want to solve: throughput of research, especially in SME biotech and the academic space, reproducibility and data acquisition. So just to summarize, we're really excited to speak with anyone who would like to alleviate their friends or their colleagues of manual cell culture. So if you're interested in seeing a demo, I can give you one. We can control our kit that's in London 5,000 miles away, which is pretty fun. But we're interested to know what workflows you'd be you'd love to have automated. So we'd love to know what ideas you have, and would be open to showing you some of the modules that we have up and running already. So with that, I invite any questions. Thank you.
Hi. Super cool. In academic labs, where basically they have time but no money, and the students are actually doing this work, how hard is to sell this to a PI? Because at the end of the day, we're saving time to the students, and unfortunately, they don't really care about that.
Yeah, and it's definitely a challenge. I think there's a number of ways of looking at it, because you can come in at a price where it's, within a personal budget, the kind of price where it's cheaper than, or it's less than the monthly spend of the consumables you're using. So in some of these scenarios, you could actually, it's quick and cost-effective enough to prototype. It's a bit like how microservices are like today with software. You can get yourself up and running at a very low cost, and then you can do an internal pitch to the rest of your team and say, look at the awesome stuff I've managed to do.
What is the cost?
So we're looking at pricing cell feed at between sort of £7,000 to £5,000, depending on what features you're after. So things like passaging and the monitoring, it'll be a bit more. But for the modules, we're looking at making them super affordable, almost to the extent where you don't even think about the cost.
Just a quick comment following up on that question. I don't think professors don't care about their students' time. They're painfully aware of how much they're paying from their grants for their students. I think one of the challenges we face in the devices we're selling them and the robotics is it going to be reliable? Is it going to work? Or is it going to take half a year of my students' time to set it up? And a lot of professors have that experience buying very expensive, we're not talking OpenTrons, but very expensive equipment that they've spent $500,000 on, and then a year of trying to get it to work, technicians coming out. So I think getting the interface, getting it to be reliable, those are the key things. But it's not that professors are unaware of the costs.
I think just to add on that as well, something that we've seen with the utilization of these modules is people look at their workflows in different ways. Instead of saying, right, I'm going to just replenish my media once a day because that's the least amount I have to do, I can now do different things, like maybe do it more continuously. So it opens up new ideas, and actually one of the motivations for that would be we can publish new methods. Publish something that hasn't previously been done before in the academic lab just purely because of the human constraint. So there's some interesting things there as well.
One thing we still haven't heard from any of these really, really cool talks in this session is the question of archiving. So let's say all of you survive and it's wonderful, and we have this blossoming of all of these different technologies and companies. But one of the things that's going to happen, I think, is all of these protocols are going to be sitting inside of your servers, right? And if and when something happens, right, what happens to all of those links from those papers? Now, I have a pretty good idea of what normally happens to links. Right about the time you publish, about 10% are broken, maybe 15%. Within a couple of years, it's more like 25, 30%. So I would love for you guys to just address that and really ask the question, what happens when you're not there?
I think that's really interesting, and I don't think I can solve that in the time I have right now. But, I mean, lunch is coming up. We should definitely all put our heads together and think about that. Cheers.
Okay. Thanks. Alicia Henn from BioSpherix. Thanks to the organizers. Thanks you all for listening when it's so close to lunch. I want to describe for you this concept that we call cytocentric. And in doing so, first I want you to think about your cell culture lab at home, those of you that do cell culture. Stand in your doorway and look around the room mentally, and what do you see? Do you see benches? Do you see cabinets? Do you see chairs? These are all cytocentric things. Your cells, people-centric things. Your cells need none of these. When you start thinking about cytocentric things, you start thinking about what the cells really need, which is unbroken physiologic conditions. Is this working? There we go. So this is a conventional people-centric lab. You can get your hands in there, you can get your face in there, it's very convenient for people, but we know that this is actually a very suboptimal environment for our cells. There's a high rate of contamination. Every time the door opens and shuts, uncontrolled room air is mixing with the cell environment, creating these variations in conditions, whether you're accessing your cells or whether someone else from the lab is accessing their cells. And you know that door doesn't open and shut the same number of times on a Sunday as it does on a Monday. So it makes it a highly variable environment for cells. When you look at if we can look at a cytocentric incubator, there we go. It opens only into controlled aseptic space that's set for optimal conditions for the cells. It's even worse when you need to do cell handling. In a people-centric lab, when you take cells to the BSC, this is cold, it's hyperoxic, it's hypocapnic, and it's highly variable environment. Every time an operator moves their hands in that BSC, their breath is literally mixing with the cell handling environment. A cytocentric cell handling space is closed. There's a physical barrier between the cell handling environment and the people environment. And we can incorporate any type of third-party equipment that you would need to take care of your cells inside this cytocentric environment. So inside this environment, you have no room air. It's all tanked gases that are medical grade. They're filtered. It's an aseptic space. The floor is heated. The air is heated. Particles are controlled. And it gives you not only 24/7 control of the environment around cells, but it allows you to record and monitor those conditions for your cells continuously. So you can take all this time that cells are out of optimal conditions. When you've taken them out of the incubator, the CO2 goes off, the pH goes off in those cells, the oxygen goes off, the temperature goes off, and after you put them back in the incubator, it's hours for them to all come back to optimal conditions. When it says 37 degrees and 5% CO2 on the outside, that's not what your cells are actually experiencing inside. When you take the cell's point of view, it's a very different reality, what's going on in your lab. In our systems, you have full-time optimal conditions for cells without this variability. More broadly, the ex vivo system allows you to protect an entire cell process, start to finish, from the point at which cells are isolated from the initial tissue through any cell handling and cell expansion that needs to be happening, all the way through to you have a final cell product or final piece of data that needs to come from those cells. So it's a totally different approach to how you look at cells and what they need to do for you in the lab. In building one of these systems, we take modules and we put them together in custom ways to create the type of cell space that your particular cell process needs in the lab. It could be a very large system, like this one that's being used to isolate pancreatic islets in a GMP system for clinical trials, or it could be a small system, like this one that's here in California, in a research laboratory. And the nice thing about having this modularity, it allows you to start small in the research setting, where you can lock down the optimal conditions for your cells when it's cheap and easy to make changes, and then you can add on functionality, add on automation, scale up, scale out, use those same modules over and over so that in the end you can have a fully GMP cell factory using the same modules that you started with and the same environment that you started with those cells early on, which allows you to avoid the stumbling block of tech transfer that comes when cells are ripped out of a research environment and put into an industrial cell production environment. Now, if I can dial in any atmosphere that I want from my cells, what am I going to pick? It's not going to be anything related to room air because that's physiologically irrelevant, right? I'm going to have physiological CO2, temp, oxygen. I'm going to go through this quickly because I'm running out of time. There's a biological basis for why constant physiologic oxygen is critical for cells. Part of it's a new understanding that we have of HIF-1 alpha, and I just want to draw your attention to the numbers here. Single minutes out of optimum oxygen conditions is enough to modulate HIF levels, which affects cell proliferation and differentiation in your final cell product. So we've worked with makers of automation from all over the world to enclose automation inside a cytocentric environment to negate the negative effects that automation has on cells, whether that's automated pipetting robots or vial filling robots or bioprinters or robotic arms for more varied cell handling. And it gives us the opportunity to attack the sort of variability we see even in the pinnacle of the cleanest clean rooms available, the highest tech cell culture environments we have, still have these same sources of variability to see in the most basic research labs out there. So when we start talking about the broader issues around reproducibility, whether it's questions about reagents, whether it's problems with reporting, whether it's problems with cell identity, all of these factors come into play to different extents depending upon whether it's one lab trying to reproduce their own results, whether it's the lab down the hall trying to reproduce their results as a collaborator, or whether it's somebody around the world picking up a report and trying to reproduce those findings. But there's one factor that adds variability to every single cell-based assay performed, and that's cell conditions. And it's not being addressed in the literature that we've been talking about, which is why I'm here to tell you today that this is very doable. This is something that we've got a good handle on. You can take these optimal conditions for cells, you can take this equipment, you can lock it down how you need it, and then clone it, and you can put it anywhere in the world. So now you have verifiably reproducible conditions for your cells, whether you're in San Francisco, whether you're in South America, or whether you're on a ship at sea. Now, what's that going to be able to do for reproducibility? I think it's going to be able to do quite a bit, because we can take this technological capability of full-time optimization and full-time monitoring and attack variability from experiment to experiment, year to year, user to user. We can take the ability for physiologic simulation and use it to attack the type of variability you see from going in vitro to animal, from in vitro to human. We can take clonable environment and this modular, expandable environment and use it to attack the type of variability we see from lab to lab, from location to location, and moving from the preclinical to the clinical. And I got to stop there, and I'll take your questions. But I'm the owner of the in vitro reproducibility group on LinkedIn, so please come join us. We just talk about all of these issues there. And if you Google Cytocentric, you'll see my blog. There's lots more info there, and I know you're hungry, so I'll stop.